The Zika virus is part of the Flaviviridae family, and its genus is Flavivirus. Some other members of this family include dengue, tick-borne encephalitis or TBE, and West Nile. These viruses are typically noticed in arthropods, mostly ticks and mosquitoes, and because of this are part of a larger group of viruses called arboviruses. Flaviviruses, however, can still sometimes even infect humans. Flaviviruses are all composed of protein, single-stranded positive RNA, and a membrane made out of lipids. The picture on the slide depicts a Zika virion. A virion is the actual virus particle that is separate from the host cell. As you can see, the RNA is encapsulated in a protein layer which is called a capsid. The protein that makes up the capsid is called the C protein. The capsid is then surrounded by the lipid membrane, which is referred to as an envelope. The diameter of the Zika virion is around 40 nanometers, and the surface projections are about 5 to 10 nanometers each. The nucleocapsid of the virion, which is the capsid of the virus and the enclosed RNA, is around 25 to 30 nanometers. The genome of the virion is around 11 kilobases long. The envelope of the virus has 180 copies of proteins M and E. Protein E is a protein dimers. That means it is basically two protein monomers combined, as you can see from the picture. Both of these proteins give the virion an icosahedral shape. To clarify, an icosahedron is a solid with 20 faces. Zika viruses are typically transmitted by mosquitoes that belong to the Aedes genus. When the mosquito sucks blood from a person infected with the Zika virus, the virus affects the cells of the mosquito. The mosquito will then develop an infection for about a week and a half, and this will cause the saliva of the mosquito to carry Zika virus particles. These viral particles can then be transmitted to any human being that the mosquito takes blood from. The transmission occurs when the mosquito sticks its proboscis, which is its nose, into the human skin. It pierces the epidermis, which contains keratinocytes that protect against the pathogens. The mosquito sticks its proboscis past the epidermis into the dermis, as you can see from the picture. The dermis is where blood vessels are located. The mosquito sucks blood from these vessels. The dermis also contains fibroblasts and dendritic cells. Since the proboscis pierces through both layers, the Zika viral particles from the saliva of the mosquito can potentially affect all the cells in those layers. The Zika particles must penetrate human cells once it enters the body, since viruses cannot replicate on their own and need host cells to do so. The E protein of the virus particle, which was mentioned earlier, can interact with receptor proteins on the cell found in the epidermis and dermis, which once again are skin dendritic cells, fibroblasts, and keratinocytes. DC sign, AXL, and tyro3 are the most common receptor proteins on those human cells that are binded to and allow viral entry. So, when the E envelope protein of the particle binds to one of these receptors, the viral particle is allowed to enter the cells through endocytosis. IFITM3 is a protein on the membrane of cells that can stop the Zika virion from binding and therefore prevent them from entering the cells. However, certain cells have low levels of IFITM3 and therefore are more susceptible to the virus. Once in the cell, the particle releases its RNA into the cytoplasm of the cell. The RNA is translated and transcribed to create specific proteins. Since the RNA of Zika is positive, it can be directly translated because positive sense viral RNA is similar to mRNA. On the other hand, negative sense RNA, which Zika virus does not have, has to be converted into positive RNA with the help of RNA polymerase, and then it can finally be translated. There are various kinds of proteins made from translating and transcribing the RNA. Some can be used to make new viral particles, and others are used to copy and process parts of the viral RNA genome. Once the necessary proteins are synthesized and the genome is copied, the endoplasmic reticulum is used to assemble new Zika virus particles. A small part of the endoplasmic reticulum is removed to form the envelope for a viral particle. Some of the proteins synthesized beforehand are used for the capsid, and the replicated portion of the RNA is used as the RNA for the new virus particle. Once assembled, the viral particle goes to the Golgi apparatus where additional modifications on the particle is done. Then the viral particle travels to the surface of the cell to be released through exocytosis. These virions can also be released through cell death. The rearrangement of the endoplasmic reticulum during this process of assembling new Zika virus particles can cause large vacuoles to form in the cell. This causes the cell to swell and die. Once the viral particles are released from the host cell, they continue to infect the other cells in the human body, continuing the cycle of infection.
The Zika virus uses a lytic cycle to infect donor cells. When the Zika RNA enters the cell, it will trick the body into thinking that the RNA from the Zika is that of the RNA produced by the cell, which would code for regular proteins. First, the viral RNA will replicate. The Zika RNA attaches to the ribosomes of the cell and synthesizes the viral Zika protein. During this translation process, the virus might also kill other cellular DNA and RNA in the way of the viral RNA. Once this viral RNA is synthesized, the Zika protein will then be broken down into different parts. Some of these different parts include the actual RNA genome, the enzymes that will copy the RNA genome, as well as the capsid and envelope proteins. These viral protein parts will then travel to the ER, where they will be synthesized into new Zika viruses. When the virus buds off the ER, it carries some of the phospholipids to help enter easier to the next donor cell it will infect, since that eukaryotic cell will also have a phospholipid bilayer. The envelope proteins also consist of receptor molecules, which the future donor cells will detect to allow the viruses into the cell. After the ER, these viruses are further modified in the Golgi apparatus, and finally sent out of the cell to infect the next donor cell. However different from a typical lytic cycle, the cell does not lyse, but rather continues to release the virus over time until enough of the cellular components of the cell are damaged, and thus the cell dies. So overall, this process represents a lytic cycle because Zika does not use the reverse dogma process to turn the RNA into DNA and then inserting the DNA into the donor cell. Instead, the Zika takes the RNA and synthesizes the proteins it needs directly from the RNA to create more of the virus. So currently there is no vaccine for the Zika virus. However, there is currently a vaccine which is undergoing phase 2 trials for patients aging 18 to 35 at the National Institute of Allergy and Infectious Diseases, the VRC 5283 vaccine. This vaccine is a DNA injection which is made of a plasma that enters the body. The dose was followed by an electroporation, where small electric currents are done at the skin at the site of injection to open up the pores into the cell to ensure for maximal DNA uptake. Muscle cells in the arm read the DNA and code for the proteins that will make the Zika virus proteins, including the capsid and envelope proteins, which will later assemble on the ER of the cells to become essentially these imitation Zika viruses. These virus particles would consist of antigens on the surface, which would then trigger a small immune response, including creating antibodies and T cells to match the virus. The virus does not spread since the imitation viruses do not contain the DNA that encodes for the enzymes in charge of the replication of the viral RNA. In addition to this vaccine, another vaccine, the GLS 5700, created by Gene One Life Sciences and Invio Pharmaceuticals, has currently passed the phase 1 trials and is ongoing the phase 2 trials. A plasma body that contains genetic code that encodes for only the envelope proteins, which encodes for the antigen shapes like the E and PRM proteins shown here. Okay, so these antigens could then activate the immune system response. The antigens would travel into the bloodstream and activate the T cells since it's a foreign item, and thus then activating the B cells to fight the plasmid. This would then result in memory T cells and B cells being produced. These memory T cells and B cells could later be used to take down the virus when it first enters the body. The body would then have the ability to quickly produce these antibodies to quickly take down the actual virus. So there is another type of vaccine. The ZPIV vaccine contains the whole virion except the pathogenic RNA part of the virus, which has been heat killed. However, it has been proven that the body can still read the antigens on the heat-killed virus and produce an immune response, again producing antibodies and memory T cells and B cells to take down the virus. If it passes phase 2 trials, this type of vaccine can be especially useful for those with weak immune systems whose bodies cannot handle the pathogenic RNA of the virus or producing foreign proteins in bodily cells. Okay, so a vaccine is necessary to prevent the spread of the virus, especially the congenital effects of a newborn, and often mesencephaly, which is a severe brain disease, can occur if a germline cell is affected. If the vaccine can immediately protect against the spread of the virus inside the body, especially in the fetus, then there is less of a chance for the germline cell to be infected. In addition, the vaccine can protect against those with weak immune systems. Many times, people with weak immune systems have bodies that cannot fight off the virus in the typical 4 to 10 days, and they may feel greater effects than the typical mild fever, headache, rash, joint pain, and conjunctivitis, and they might feel these effects that are shown here. So overall, the Zika virus is a part of the Flaviviridae family and is made up of a single strand of RNA and the proteins which enclose it. The virus is transmitted when an infected mosquito pierces a human skin, thus infecting the human with the virus. The life cycle of the cell allows for the virus to quickly spread around the body. This virus typically affects 1 in 5 who have been infected. However, a vaccine is necessary to protect mainly pregnant women to protect against congenital effects of the baby if germline cells are affected. A DNA vaccine is now undergoing phase 2 trials to protect those who may suffer the effects of a Zika virus. And these are our sources. Thank you.